But for the new volunteers, while we're going through this PowerPoint, please pay attention to the text that is in bold, because that is key information for you to know to pass your certification quiz. So with that, let's go ahead and get started. Okay, so for those of you who are unfamiliar with our program, we are Georgia AdoptAStream. We are the state's citizen science water quality monitoring program. It's a little bit of a mouthful, but we encourage citizens to get out and to familiarize themselves with their watershed, monitor impacts to their waterways, improve streams, rivers, wetlands, any body of water near them, and inform others about the effects of water quality throughout the state. So AdoptAStream has 5 goals, which can be memorized using the mnemonic device ADOPT. So I'll go ahead and go through this right now. A stands for awareness. So our program, we strive to increase public awareness of water quality issues throughout Georgia and the neighboring southeastern states. We do this through outreach events, through monitoring. We do it by providing accessible data through the database, and by also housing this network of volunteers to exchange information and to support one another. D stands for data. Kind of like how I mentioned before, we have an online open access database that houses thousands of data points. So anyone can see this data, and over time, the baseline data that's collected by volunteers shows natural fluctuations and trends in the water bodies that they're monitoring. And by monitoring these sites, you can see any deviations over time, and these can indicate a possible pollutant or impairment to that waterway. O stands for observation. So an incredibly important part of monitoring is being aware of the area around you and making careful observations. So just by noting what you see or what you smell at your site, like a sewage spill or another pollutant source could be found way before you even start taking your first sample. So it's important to note any changes that you see over time, because these represent impacts on water quality as well. Going on to the next one, P is for partnerships. Forming partnerships is very important throughout your journey as an AdoptAStream volunteer. So not only do these relationships help you get equipment that you need, but it'll also give you the support in case you find any pollutants or any harmful impacts to your site. We all have something to offer when we work together to solve problems. So we encourage partnerships between volunteers and their local government. And finally, T is for tools and training. So that's what you're part of today. After you complete this presentation and then the second part of the certification, you will have the knowledge, the skills, and the tools to monitor the bacteria within your site. So AdoptAStream has many partners that we work with to help provide our volunteers with the equipment, support, and the training opportunities that they need. All right, so why are we here today? Why do we want to monitor our waterways? Why are we monitoring for bacteria? So before we can answer any of these questions, there's a really important term that we need to know first, and that is a watershed. So a watershed, if you do not know, and pay attention to the bold text, like I said before, a watershed is an entire area of land from which all of the water, sediment, dissolved materials flow and drain to a common point, i.e. a water body. So the boundaries of a watershed are defined by the highest points of land surrounding that water body, and everything that touches this land, so that includes plants, animals, pollution, any man-made structures or disturbances, all of this impacts that water body. So this is where we start to see the cumulative effects of non-point source pollution and potentially the harmful impacts to ecosystems that also impact human health. So essentially, each water body has its own watershed, and the size of a watershed will depend on the size of that water body. So it can be as small as a neighborhood creek, or it can go, be as large as a river that goes across the entire state. So now that we have an idea of what a watershed is, where can they be found within our state? The answer is literally everywhere. Everything and everyone is located within a watershed. So Georgia has 14 major watersheds. You can use this map. It's kind of small on my screen, but maybe it's larger for you. You can use this map to find your county or your area and just pinpoint what watershed you're in today. So feel free to put in the chat where you're tuning in from and what watershed you're in. I'm in Kennesaw right now, so I'm actually on kind of the boundary line of the Etowah and Chattahoochee watersheds, but I lie more in the Etowah. The second photo over to the right shows the 52 sub-watersheds of the state. So the key thing to take here is that these larger watersheds can be broken down into smaller watersheds. And even though these land areas are separated by high points of land and other factors, it's important to remember that we all live downstream from one another. So the actions upstream will affect the quality of water of those downstream. On to the next slide. Thank you. All right, this map here, this shows all of the active AdoptAStream sites across the state. So working off from the previous slide, there might be some volunteers working in your watershed already. And if that's the case, then there's already, this network of volunteers that you can join and, gather information and resources and support from them as you begin your monitoring. And if you don't see any sites in your area, then that's great. And we're excited to see you here today because you're leading the way in your area. And we're thankful for you. So by becoming a part of AdoptAStream, you are joining a network of volunteers across the state that are collecting and submitting data, every month or every season. So these can be teachers and students that are collecting data for research purposes. It could just be an individual who has an interest in water chemistry. It could be a family that is interested in knowing the health of their backyard stream. Our volunteers are diverse and the reasons why they're monitoring is diverse as well. So it's a huge network of volunteers that you're joining. And to kind of show the structure of our program, we have this pyramid here. So at the base of the pyramid that provides really the biggest support in the hands-on, boots on the field are the volunteers. So they are the reason that we exist. Without such a large number of interested and active community members working as AdoptAStream volunteers, we wouldn't be able to monitor close to the number of sites that we have right now. So thank you to all AdoptAStream volunteers. Because of you, our database is ever growing and we're here to support your efforts. The next level up the pyramid are the local coordinators. So local coordinators or trainers are really important because they provide the training and support to regions throughout the state. And it's their role is to stay up to date with the information that the state and local governments put out. So these coordinators are trained by the state staff and provide many of the workshop opportunities that are being offered right now. And then going up one more level, we have an advisory board that guides the program to be the most effective and meaningful for the citizen scientists that we engage with. And then at the top of the pyramid is just the state staff. So, you know, we are the smallest team here. Right now, we are a team of, I have to think about it, just 4 of us, right? Just 4 of us. So, you know, we help provide the information, the support, and the guidance whenever possible. And, you know, we also organize an annual conference every year to bring together our network. And we also do a lot of traveling to help participate in other outreach events and data collection events whenever possible. So, you know, you just saw we have so many volunteers and coordinators throughout the state working with the program, but we all need to make sure that we're on the same page with monitoring, just so we know that our data being collected is replicable and trusted. So AdoptAStream has an EPA-approved Quality Assurance Project Plan, or QAP. So the QAP basically lays out all of the details for quality assurance, quality control, or QAQC protocols. These QAQC protocols just simply give every monitor, every volunteer, a standard to abide by when they're monitoring. And once we start talking about the actual bacterial monitoring this evening, then you'll see that there are steps and processes that every volunteer should follow. And these steps are the same for everyone that submits data to the database. So besides providing protocols, the QAQC framework just provides a baseline for volunteers to get certified, to submit data, and continue working with AdoptAStream. So before you become a certified volunteer, you have to attend a workshop first and pass the certification. It's called a test, but it's not like a test, it's more like a quiz. But you know, you do have to attend the workshop, attend the field portion, and pass the certification quiz. Only individuals are certified. So this means that if a teacher gets their certification, then only the teacher has the certification. The whole class, the students, are not certified because it only goes to the person who attended the workshop and passed the certification exams. All right.
Awesome. Thank you so much, Jackie. Now I'm going to kind of take us into the more focused part of the bacterial presentation. So Jackie just gave us a lot of really helpful information about, you know, our program and kind of what it takes to earn a QAQC certification. And like she mentioned at the beginning, this presentation is kind of just the first part of actually earning that certification. So by attending this today, you kind of got that first part under your belt, but then we're going to end up connecting you with a local coordinator or meeting up with us so that we can complete that field and lab portion, which you see kind of photos of here on the slide. And then we have that written test, or as we like to call it, written quiz, because test seems a little intimidating, but you have all the information you need that is here in the PowerPoint. And so I'll go ahead and go to the next slide, which has some bolded information, which, you know, is there for the test so that you know it and you have it all there and you're prepared. Yeah, so we've kind of talked about all these things, and so now we know how to get the certification, but why would we actually want to get that certification to start monitoring? Well, monitoring for E. coli can be extremely informative, as it can indicate when human health and potentially ecosystem health are at risk. So by doing this and monitoring this bacterial contamination of surface waters, we're assessing if pathogens are present. So pathogens, that's really just any microscopic organism that can cause disease. We'll go into a little bit more detail on the next slide, but that's just something to remember there. this type of monitoring that we're going to be talking about and kind of talking through is going to involve collecting some samples, plating those samples, counting them all out. And so you'll get to see that here. But this is really helpful, and like we're saying, assessing these risks pretty quickly, and just getting a snapshot of if there's any contamination. So this E. coli sampling is not going to tell us a long-term trend necessarily. When we go out and grab that sample, we're just looking at what's kind of contamination is going on in the stream right then. So we can't really infer and say, well, you know, this time next year, my levels will be this. That's not really exactly how this protocol works, which hopefully you'll be able to understand as you get into the info a little bit more here. All right, so we talked about sampling for bacteria, but what are bacteria? They're just single-celled living microscopic organisms. So bacteria do have a pretty bad reputation sometimes, right? Especially in the world we live in now, with all the germs we feel like are floating around. And there definitely are bacteria that cause diseases, and these are just one type of pathogen. There are also different types that release toxins and can cause different types of harm to humans, to ecosystems, wherever it may be. But we would be remiss not to mention all the good things that bacteria do for us too, because there are more bacteria on Earth than pretty much any other living thing, and they are found in every single environment that we have scientifically discovered thus far. I think bacteria have been in every single one almost. So we can't go without saying there are some pros and good things that they do. Bacteria help with decomposition. It's an important part of the life cycle, breaking down organic matter and nutrient cycling. We're returning nutrients to our soil and to our atmosphere. We're making sure that things are healthy and we're keeping up with those natural processes. Bacteria are responsible for part of those things. Digestion, something that we couldn't go without, right? Our gut microbiome and the bacteria in our stomach help break down food, which is really helpful for us and other organisms. And then we have pollution control. So maybe you've heard about some different bioremediation techniques where bacteria are used to kind of clean up maybe oil spills or other types of substances that could be harmful to different environments. So yeah, this is just kind of like broad overview of bacteria, but now we're going to kind of focus in a little bit more on the types of bacteria that we're going to be specifically looking at. So we kind of have this little chart here. We can see that the largest circle is coliforms. So this is a biological family, as you can see here, of bacteria that are naturally found in the soil. And then we have fecal coliforms, which is kind of a subgroup of those coliforms, and these are found in intestinal tracts of warm-blooded animals. And then that will in turn come out in waste product. So one type of a fecal coliform is E. coli. So this is a smaller subset within fecal coliform, and it makes up about 60% of the fecal coliform group. So this specifically, E. coli, is what we're going to be focusing on throughout the rest of the workshop. So we know that monitoring for E. coli is pretty important because it can tell us quite a bit, but why specifically E. coli? Well, E. coli is actually an indicator species for pathogens. So maybe you've heard that term before, indicator species. But really, that just means it's a species. If we find it in a particular environment, then that means it's going to indicate that the environment is conducive for other pathogens to live there. So if we're finding those, then there's most likely going to be other pathogens in the area that we would want to be aware of, because like we now know, pathogens are things that cause disease and cause harm. So we want to be aware of those. Excuse me. So And also it's not really practical to test these waters, especially for volunteers, for all these different types of pathogens, when really we can kind of focus in on this one and it can give us a lot of information. So high levels are going to tell us that pathogens are present. But how do we get to high levels? How does E. coli even get into the waterway if it comes out of, fecal matter? So unfortunately, it turns out quite a few ways that E. coli can get into waterways. And if it's coming out of warm-blooded animals, right, it's definitely going to be something that we can impact as warm-blooded animals. And so that would be, you can kind of see sewage here on the slide has lots of little bullet points under it. Excuse me. And that just kind of goes to show you that there are quite a few ways that sewage can get into streams and rivers and different water bodies. So maybe this urban stream runoff too is kind of another way. If you're having pet waste that maybe is going to get washed into rivers, different things like that when you have a big storm. And those big rains can also sometimes contribute to these sewer issues, maybe a combined sewer overflow, things like that, where we're getting those sources of contamination in our waterways. But we also have, as you can see here, our lovely bovine friends in this stream. We have livestock, or maybe there's, you've seen chicken farms and things like that before. So any of that fecal matter that will eventually, as Jackie told us about watersheds, everything's eventually going to go back to that stream, to that common point. So there's also another way. And then we have wildlife too, because wildlife's just out there living their life and they need to be able to do their business out there too. So that is also going to impact our waterways and how E. coli gets in there. All right, so these are some different factors that will influence the E. coli counts that we're getting. So we know how it gets in there in the first place, but these are some things that can contribute to higher numbers. One of them is weather. So if we have a big rain event, or maybe it's not even big, you just have a rain event and you're in an urban area with lots of impervious surfaces, we're going to have all of that runoff. And that runoff and that rainstorm is going to carry different types of pollutants, specifically fecal matter, and E. coli will be then present in the stream after those events. So we always like to say avoid sampling during a high flow or after a heavy rain, because the flow can be kind of dangerous, but also because you're pretty much guaranteed to find contamination after that, no matter where you are. But outside of weather events, we also have the seasonal changes that we need to be aware of and different types of temperatures, because temperature definitely affects the types of E. coli levels that you're going to find. So when we have warmer water or like warmer surface water temperatures, that's going to in turn make E. coli replication rates go up. So E. coli like nice, warm, cozy environments, and when they're nice, warm, and cozy, their replication rates go up. But when it's colder, their replication rates decrease. So that's something we can keep in mind too if we see any kind of seasonal maybe fluctuations, or maybe just times during the day when maybe it's hot in the middle of the day, but it's a little bit cooler in the afternoon. So that's something to think about when you're actually out there monitoring. So when to monitor is a big one that we talk about in the same time of day because of the thing we just talked about, right? That temperature can definitely impact the replication rates, which will then in turn impact the counts that you're going to end up finding. So also normal flow conditions, which we just talked about, but those are good things to keep in mind. But then where to actually monitor. So if you have a stream site already, with AdoptAStream, that's great, and you can just incorporate your bacterial sampling in that site. But if not, definitely choosing one site and kind of going back to that same location every time is going to ensure that we get kind of this replicable data and comparable data. So that's important. And then we want to make sure that the area we're selecting, we have well mixed and flowing water. So be careful that we're not choosing an area that's kind of stagnant or we don't see much movement there. We want to definitely look for flow. And when we are grabbing your samples, we just want to make sure we're not getting a bunch of sediment in our bag and different things like that. But that's something we'll talk about, or Jackie's going to tell us about here in just a minute when we go through how to collect our sample. So if you're interested in, kind of adopting this bacterial routine into your AdoptAStream monitoring, then this is something that we recommend is done once a month. You're a volunteer, so we appreciate any time that you contribute to our program. It can be more or less, completely up to you, but we do recommend once a month for this sampling type. Now I'm going to hand it over to Jackie so she can talk us through these steps.
Okay, so let's get into the 5 steps that we do to monitor for E. coli. So step number 1, we want to prepare our blank or the control. So a blank is your control sample, or it just proves that there's no contamination occurring during your sample collection, transportation, or plating. So the blank is actually just prepared with distilled water, and it is held under the same conditions as the samples from your site. Just the biggest difference is that it has the distilled water in it. There should be no bacteria of any kind present on your blank once you incubate it and grow it. And if there is bacterial colonies on there, then there has been some form of contamination. And if that happens, then you must discard all your samples and redo the sampling process, because the whole point of the blank is to have no growth whatsoever. So how do I prepare my blank? You're going to use a Whirlpack bag. So these bags are sterilized, and they're what we use to collect our water sample for bacterial monitoring. You can write on them. So you just, with a permanent marker, you can label the bag with the word blank on it, the date, and then the time that you're preparing this. So you can see here on the photo on the right, an example of that. And then you're going to want to put on gloves, because we want to make sure everything stays sterile. So you're going to put on your gloves to pour the distilled water into your bag. Put on your gloves. There's a perforated seal at the top of the bag that you're going to remove. And remember that this is a sterilized bag, so we don't want anything to get inside it except the distilled water. So there's small white tabs on the outside on the top that you'll use to pull it open. And then you're going to fill up the bag about 2/3 of the way up with that distilled water. Make sure that the bottle or the container of the distilled water doesn't touch the inside of the bag either. And then you're going to whirl it. So unfortunately, I don't have a Whirlpack with me tonight, but you'll just hold it by the yellow tabs. You can see it in the photo, and you're just it's a whirling motion with your wrist. And that's how it seals it up. And then it's just kind of like a twist tie situation with the yellow tag ends. And that's how you seal up the bag. Then you're going to place your blank into a sanitized cooler with ice. And remember, this is going to be the same cooler that's going to hold the other samples from your monitoring site. So you only need 1 blank per cooler, but you can have samples from multiple sites in that same cooler. We just like to note that in case volunteers are monitoring more than 1 site. A quick note, you can prepare this blank at home, but if you do that, then you should immediately place it in the same cooler that you'll be taking out to your site, or you can prepare the blank out in the field. However, just keep in mind that all the samples that you collect including the blank, must be processed, plated in the incubator within 24 hours of collection. So just be mindful of that, whatever way you decide to do, but it'll probably make more sense as we go along further with the steps. So step number 2 is going to be actual collecting our sample. So, you know, we have our blank prepared. Make sure you take the cooler out with you, but time to head out to your site. So with your WorldPack bag, you're going to label it with the location name, your site name, the date, and the time you are collecting your sample. You're going to use the same procedure as you did with the blank to open up the bag with the gloves, making sure you don't get anything on the inside of the bag, trying to just avoid any cross-contamination. And you're going to fill your WorldPack up again about 2/3 of the way with your stream water. Ooh, thank you, Gretchen, for pointing that out. This volunteer does not have gloves. I can let Bailey address that, but yes, very important just to wear gloves because you never really know what might be at your site. But you're gonna collect the WorldPack sample facing upstream because anything downstream from you is gonna be impacted from all the disturbance that you just created getting into your stream. And then you're going to collect the water, holding the yellow tabs on the side, just scoop it up. This is what we would demonstrate in the field during the in-person session. So this would make more sense then. But once you have your sample, then just give it a whirl away from your face and then seal it up, put it in the cooler with your blank. So remember how we said that the temperature is a really important factor with bacteria. The cooler temperatures help preserve the sample and prevent the breeding. The higher temperatures breed bacteria. So it's important that you have ice in your cooler. You also want to make sure it has a lid because the UV rays, exposure to the UV rays will kill any of the existing bacteria in your sample. And then you'll just want to make sure that you plate these samples within 24 hours that you collected them. On to step 3, actually plating your sample. So you want to make sure that you clean your area with disinfectant beforehand. And then you're going to invert the WorldPack samples just a couple of times to mix it up because settling does occur. And you'll be using 3M Petrifilm plates. These are plates that have a special medium on them that, it's designed to detect E. coli colonies. So these Petrifilm plates must be kept in a cool place, and they also have an expiration date on them. So you'll want to make sure that they're not past that expiration date. You're going to have 3 plates for each sample plus your blank. So you can see here in the photo, there is the top plate is labeled blank, and that's where you're going to plate the water from your blank bag. And then the 3 plates on the bottom are the 3 plates created from your one site bag. Hopefully that makes sense. But you're going to label these plates with the date, the time that you're pipetting it, creating it, the site name. And you also just want to make a note if it's the blank, if it's plate number 1, 2, or 3 of this site. So you're going to take the pipette to draw out 1 milliliter sample, and then you're going to just pipette it onto the plates. And during the in-person field portion, you actually have hands-on practice to do this because you just want to make sure that you're pipetting it right nicely into the middle of the medium. And then when you're putting the cover back on, you want to just do it nice and slowly so that you want to avoid getting water off of the plate and onto the table. Okay, step number 4, we're going to incubate our plates. So you'll use an AdoptAStream or EPA recommended incubator to do this. We recommend that you beforehand that you kind of play around with your incubator and get to know it, because they tend to be temperamental sometimes. So, you know, play around with it beforehand before you actually go and start collecting your samples, because you're going to need to preheat this incubator to about 34 to 36 degrees Celsius. And you're going to place your plates into this incubator to incubate for 24 hours, give or take 1 hour. You want to make sure to check the minimum and maximum temperatures after you incubate them, because you want to ensure that your incubator didn't go outside that range of 34 or 36 degrees Celsius. So that's why we recommend that you get to know your incubator, just in case it's a little temperamental and you're going to have to, you're going to have to continuously check on it to adjust the settings or the temperature. And step number 5, thank you. Step number 5 is going to be reading our results. So 24 hours have passed. Our plates have these different colonies growing on them. We're not sure what we're looking at. Well, the E. coli that we're counting are the blue colonies with gas bubbles. And I know this is a black and white photo right here. We have some real photos coming up to get you some practice. But this little graphic right here is just to show you examples of what possible gas bubble patterns might come up on your plate. So the E. coli that we're counting are the blue colonies with gas bubbles. There will be red colonies that may also appear, but these are just general coliform. So we are not going to count those for our monitoring purposes. We may also see just blue colonies without bubbles, and we also don't count this because these are just not viable colonies. Additionally, another good point is that you don't want to count any colonies that are more than halfway off the median. So we'll practice with that here in a moment. And then our final units for bacteria, when we're reporting this, is going to be colony forming units, or CFUs, per 100 milliliters. And keep that in your head for later too. We'll go over it in a minute. But I'll hand it over to Bailey so we can practice some plate counting.
Yeah, thanks, Jackie, for getting us through the whole procedure. Yeah, so now we're going to ask for a little participation, I guess. We'd like you guys for this next section to you're welcome to unmute, but also if you just want to use the chat to kind of throw in your guesses of how many E. colonies, E. coli colonies, we're going to get some practice going through and counting and picking out the E. coli versus just those regular coliform, which is kind of that distinction Jackie just told us about. So but before I get ahead of myself, we're going to take a look at the blank. So this plate is what your blank or your control sample should look like after it's come out of the incubator. So like Jackie said, we've already been, it's been 24 hours, well, I guess 23 to 25 in the incubator. You pull it out. Your blank should look like this, and there should be no colonies growing on it at all. And if there were colonies growing on it, then what would we do? We have to do everything all over again, right? And it's not necessarily like oh, quick, I'll just go right back out and do it again. It is, as you can see, like a 24-hour at least process. So that is something to keep in mind. And that's why we have some folks chiming in the chat about using gloves and making sure that we're following the protocol as closely as we can because it's pretty easy for contamination to kind of hop in these different stages. So we want to be aware of that. All right, so now we're going to get into our practicing here. So I'll just give everybody, I don't know, a few seconds. I was going to say minutes, but minutes would be too long. A few seconds here to just take a guess, look through on this plate and put into the chat what your guess would be for how many E. coli colonies or those colony forming units we can find. Okay, I see the chat. Let me pull it up. All right, we got 2 answers. Okay, we got 5, we got 4, 5. Awesome. 5, 6. Okay. Great. Thanks, guys. So now we have this handy dandy new thing in our new PowerPoint where it's gonna hopefully show you the ones that we have circled and identified as those E. coli colonies. So we can see here what we're looking for again are going to be the blue colonies with a gas bubble, right? So we can see all of these other little colonies on here, and they do have gas bubbles, but that's what Jackie explained are those red colonies. Excuse me. And so those are just going to be kind of like our general coliform, which is not what we're looking for, right? We're sticking with that indicator species of E. coli. And so thanks everyone for chiming in. You did pretty good. Everyone got really close. I will go ahead and let you know that it's much, much easier to actually read these plates when you're holding one in your hand, fresh out the incubator, and you're gloved hand, right? Fresh out the incubator. It's much easier to see that colored difference and the differentiation and kind of this, you can kind of see here, well, I was going to try to show the pointer. It's like a blue smear almost where the plate is designed to color those E. coli colonies. So when you see the smear, that's a good indication. So we've done our best to update the photos and get as high resolution as possible with good lighting, but there's nothing like the real thing, as they say. So just keep that in mind as we're going through these. So here's our next one. All right, what are our guesses for this? Awesome, thanks everyone. We got 2s, maybe a 3 in there. Okay, all right, let's click here. So on this one, we have circled 2. This one kind of does look dark, but we're not seeing that kind of smear like we can see on these other ones. So that's the reason we did not count some of these other larger colonies where you can clearly see that gas bubble. So for this one, you would say 2. And I'll go ahead and let you guys know too. So Jackie told us that we're running in triplicate, right? So having 3 plates per sample. All 3 of your plates are generally in a pretty tight range of kind of the same number. So if we had this, we can kind of expect all of our plates from that sample to be between like, maybe 0, 1, 2, 3, 4, maybe 5, but I would expect them to all be, pretty close there together. So that's something to keep in mind. It's a little bit easier. You're not hit with like 2, 1, 7, 90, we're not going to have all of that. They're pretty tight in range. All right, so what do we have for example 3? What would you guys guess? Oh, y'all are fast counters. That's awesome. All right, we got 9, 11, 12, 13, 11, 11. Okay. Awesome. Okay. Oops, I X out the chat. There we go. All right. So we can see here, I think we have about 12. We have 12 circled on this one. And then we just want to remind folks, we're not counting it if we don't see an associated gas bubble. So you can see some kind of gas bubbles maybe like floating around it, but we also see other, those general coliform colonies. And we're thinking that those gas bubbles are associated with those different colonies. So if there's not a clearly associated gas bubble, then we're not going to count them, right? That's what Jackie told us when we're looking for these viable E. coli colonies. So good job, everybody. We got a really close range on that one. All right. What's this one? Yeah, Gretchen went ahead and put it in there. TNTC. So what does that mean, TNTC? Too numerous to count. So pretty obvious, right? That, I mean, if you were to see this, you would be like, oh, no way can I count every single one of those little dots, right? And so this is a good thing for us to bring up right now, because I'll go ahead and click this next one. So as you can see here, this kind of like, we put this little photo in the corner as kind of a reference, because we like to talk about how when you get too numerous to count, you're obviously going to see a lot of colonies on the plate, right? We are also going to see the plate change color. So we have this kind of reference photo down in the corner to show you this is what a plate would look like, you know, with its typical kind of colonies. I think there's maybe like, you know, 6 on there or something. But when we're looking at TNTC plates, we can see that the background has kind of changed to this dark pink or maybe purplish color. And that's going to tell us, or that's going to give us a pretty strong indication this is probably a plate that we can label as TNTC. But another thing would be, we can look at it and we can definitely see that there's some blue smear here and there, but it's very hard to differentiate which what color difference is and what exactly you can count as a blue colony versus a red. So those are some good indicators of when you would actually label a plate TNTC. I think the people we buy our plates from, which is 3M, they generally say anything higher than, I think, 150. But hopefully you wouldn't have to count to 150 before you were like, you know, this is TNTC. So just use those few little indicators to help you get to that point. All right. This slide, what are we guessing for this one? Two. All right, 2, 3, 2. Okay. Thanks for sticking with us, guys, and doing this ID portion with us, for chiming in. We appreciate it. All right. So got some pretty tight guesses in there. For this one, we put 2. So maybe the one in this kind of far left right here is maybe something that tripped you up. It kind of tripped me up at first, but we're not seeing that smear again, where we can see here kind of like the blue radiating out from the colony. It's just a bit darker, right? But this is another reason for us to bring up, as long as we're reasonably close in our counts, we're not way off, then our calculation is going to kind of take those potential human errors into consideration. All right, example 6. What would we guess for this one? Man, y'all really are fast. Awesome. Okay, we got 10, 11, 10, 9. Again, we're in a really close range, so that's really good. Awesome. Another 10, great. So I'll show you the ones we've circled. Excuse me. We have 11 circled here. But again, it's hard to see over a screen, especially since I'm sharing my screen and then you're viewing it through a screen. So we can take those things into consideration. But yeah, those are all really tight numbers, a tight range, so that's good. All right, what do we think for this one? Example 7, yeah, TNTC, good job, everybody. Absolutely. We can see the color change. Yeah, Michelle says, TNTC, we have a problem. That's exactly correct. Yeah, so that's again too numerous to count. All right, so this plate, how many colonies do we see? Awesome, yeah. Everyone is putting in the chat 0 E. coli colonies. Good job. So then we have a question. Would this plate be an acceptable blank? Right, we've got people chiming in the chat. No, definitely not going to be an acceptable blank, right? Because remember that first plate we showed at the beginning, there are no colonies growing on it at all, and that's what we want. We don't want any colonies, even though technically there are no E. coli colonies, right? We're still seeing bacteria that's growing and thriving on this plate. So we want to make sure that if this happens, we're going to sample again because there's some kind of contamination. All right, example 9. We're nearing the road here. I think there's only like 11 or 12 examples, so we're almost to the end. All right, great. We've got 12, 11. Okay. All right, so on this one, I think we have about 13. Again, here we can just kind of see like some of these are just pretty dark and it's kind of hard to differentiate, but this is another good one for us to kind of point out right here. We can see the edge of the plate, right? It's kind of this dark pink line. I don't know if y'all can see my mouse that I'm pointing out on the screen, but we have this dark pink line that's kind of the outline of the plate. So when Jackie was telling us, we're not counting anything halfway outside of that medium. So anything that's more than halfway outside of that line, we're not going to count. But we did count this one because you can see the line here and it's definitely more than halfway inside. So that one is one that we counted. All right, example 10. I'm not going to make you guys count this slide. If you really want to do it, go ahead. But I will say this is a slide that you would count if you got it out of your incubator. Excuse me. But we're not going to try to count it on the screen today. But we like to bring this slide up because, again, here we're going to kind of throw in this little reference plate to show, again, there's been a change in color, right? It's not as distinct or maybe as dark as the color change that we're going to see if our plate is TNTC, too numerous to count, but we've definitely seen a shift in the color here. So it is a bit of a darker pink, but we can see that it's a lot easier to differentiate what's actually a blue colony and what's a red or excuse me, a coliform colony. So this is kind of an exception to the rule. I will preface what I'm about to say with this is not something you're going to see regularly, at least we hope not. Most people don't encounter this on a regular basis at all. So, but this is an exception to the rule that Jackie told us at the beginning that was we're not going to count blue colonies without a gas bubble. Okay, And the reason why for this situation where we're seeing the color change on the plate is a little bit darker pink, we can still differentiate what's blue from what's red, is because on this plate, there are a lot of bacteria, obviously, but there's not enough food, which is kind of what the Petrifilm plates are serving the purpose of. And so it can't support all of the colonies, so not all of the colonies can form a gas bubble. So 3M, which is the maker of Petrifilm, this product, has advised to count all blue colonies as presumptive E. coli colonies, whether or not they have a gas bubble, only in this situation, right? I know that can be a little confusing. We're saving questions till the end, but we can definitely come back to this slide if there are more questions. And that's just kind of like the exception to the rule. We're not going to try to trick you on the test with that or anything. So if you ever have any questions when you're out monitoring, snap a photo, send it to the state staff, send it to your local coordinator, and we're very happy to help you out. All right, I know I'm taking long on this ID section, but we're almost done. This is a quick one, hopefully, right? How many E. coli colonies do we see? Two. Awesome. I have people asking, what's the answer for the number of colonies on this last slide? And we counted about 60-ish on there. It's a little hard to see in a photo. We also counted them via the photos. So that was our guess. But yeah, everybody did great on example number 11. We had 2 E. coli colonies present. All right, I think this is the last one. How many E. coli colonies do we see here? Great job. Okay, we have folks chiming in with 3, and 3 is correct. We see those 3 blue dots, all with gas bubbles. So great job. Thank you so much, everyone, for helping us counting those. We appreciate your participation and chiming in this evening. Jackie's now going to talk us through what calculating our results will look like.
Right, so we won't have you count any more plates right now. So we just went ahead and put the counts into the table here along the bottom for each plate. So this table, it's very similar to how the data form is set up. But plate number 1 has 5 E. coli colonies, plate number 2 has 2 colonies, and plate 3 also has 2 E. coli colonies. Now let's figure out how we calculate our results. So first thing you're going to do is calculate the average number of colony forming units per slide across your 3 slides. Okay, and remember that each slide only holds 1 milliliter of water. So you're gonna add up the counts for all 3 plates. Bailey, if you can go on to the next one, awesome. Oh, sorry, I'm thinking ahead of myself. But you're gonna basically add up the counts for each 3 plates and then divide that number by your total number of plates, which is 3, to get your average. And then because our final result needs to be in average CFUs per 100 milliliter, we need to multiply that average by 100. So that shows you how to do that in step number 2. So let's try it with our numbers now. So we're going to take the average of the plates, 5 plus 2 plus 2 is 9, divided by 3, you're going to get 3 CFUs per 1 milliliter, but we need that to be in CFUs per 100 milliliters. So multiply that by 100, we get 300 CFUs per 100 milliliter as our final result. All right, so what do we do though if we have the 3 plates saying TNTC, TNTC, TNTC? how do we calculate that? Well, it's going to take a lot of math. Just kidding. It's not going to take a lot of math at all. It's very simple. Your final answer is going to be TNTC. So, you know, of course, we don't want our samples to ever come out like TNTC, but it's pretty simple to report the final result when it's like that. I will make a note that you should never have one of your plates be TNTC, but then have the other two be a count like 5 and 4, okay? If that does happen, then you should disregard these samples and test again. So you're gonna want to redo your samples and also report this to your local coordinator. So again, you shouldn't have like one plate TNTC and the other 2 plates be, you know, 6 or 4, whatever it be. But moving on to our 7th protocol is disposal and cleanup. So, you know, wrapping up, we've counted, we've calculated. We want to make sure we disinfect our working area and dispose of all of our materials. So the sample water from your Whirl pack could be, you know, dumped down the drain or in the toilet. You can also use it to water your plants. But the plates themselves, keep in mind, they are growing live bacteria colonies. So these need to be sealed either in a Ziploc bag or in a used Whirl pack. Seal it up, or sorry, spray it with disinfectant first, seal it up, and then you can throw it away. You also want to make sure to disinfect your incubator, all the surrounding surfaces that you use, and most importantly, wash your hands. Even if you did use gloves, please wash your hands. So, very important. And then our petrifilm. So I mentioned before that petrifilm needs to be kept cold. You should either keep it in the fridge if you plan on using it within a month, or if not, then you can store it in your freezer and then just thaw it before you use it. When you're storing it and you're going to use it, make sure again to pay attention to those expiration dates. All right, and then I think Bailey, you got this one?
Yes, sorry, I couldn't find my unmute. I'm here. All right, thanks, Jackie, for talking us through the calculations and kind of wrapping up all of the procedures. So now we're just going to talk about the results and what those results actually mean, how we can interpret them, and if we need to take action. So this is something that's relatively, pretty new. For us that we're presenting today, EPA and the state of Georgia. So the state of Georgia switched from using fecal coliform bacteria to evaluate the risk of illness from fecal contamination to using E. coli. And that happened just this year. So that's really exciting news, and we're happy to relay this and talk about it. Because first of all, it reflects the established EPA guidelines, which you can see kind of in this table below. But now also this regulatory data can be directly compared to AdoptAStream data. Whereas before, some calculations needed to be done in order to compare, fecal to E. coli. But now we're going to be able to just directly look at that comparison, which is really great. It gives us kind of some actionable steps so we kind of know what these things mean. These new state standards also include enterococci to indicate the presence of pathogens in coastal waters. So coastal waters are a little bit different than freshwater in a lot of ways, but also in a way that we're looking for bacterial contamination. We're going to be doing a different protocol. So you have that value there on the slide, but we're just going to assume that most folks here today are looking and working with fresh water. So that's what we're really going to focus on, since all of our methods are also focused on looking at E. coli, right? So Jackie kind of showed us what the calculations looked like, and we saw on that first calculation that she walked us through, if your plates have an average of just three colony forming units, that's going to get you a value of about 300 colony forming units per 100 milliliters. So hopefully everyone can remember that. But that just kind of shows you, man, if I only am finding an average of 3 colony forming units on my plates, look how close I already am to this regulatory level, right? So it doesn't take much for this E. coli and these pathogens to be of kind of a level of concern. So that's why it's also really important for us to go out and check out these levels. So we do have some kind of new terminology that we're going to talk about in terms of these regulatory levels. Don't want this to confuse anyone. The main number we're going to remember is 410 colony forming units per 100 milliliters. That's the level, the state standard, the EPA recommended level for E. coli. And that's based on a risk level of 36 out of 1,000 people getting sick. And those are from primary contact activities. So that's going to be like swimming, anytime that maybe water, you're going to ingest it or get it in, your eyes or anything like that. But this value is also a statistical threshold value. So what does that mean? Don't let the mathematical terminology frighten you. Really, that just means that no more than 10% of your samples should exceed a certain value. And the certain value in this case is that 410. So therefore, if we're kind of looking at it, we, in terms of like bacterial monitoring for AdoptAStream volunteers, if your samples over a year, and remember we're recommending that you sample every month, so that's 12 samples out of the year. If you take 10% of 12, that's like 1.2, right? So if you have 2 or more months out of the year where your samples or your reports are above that 410, that's something that we need to be concerned about because it's outside of the regulatory level and the regulatory parameters. So that's something that maybe you just can keep in mind. You can kind of have a reference for what these levels actually mean and what maybe the threats pose to you and your ecosystem. But I mean, this is kind of the long game we're thinking about, right? In terms of like this regulatory level. If you get above 410, maybe sample again. But we're really just looking as it consistently above. So if y'all have any questions about that, we'll take them at the end because I know we're running close to time here and I want to make sure we get enough time here to finish. But yeah, we're excited to kind of not really reveal, but talk to you guys about that new regulatory level and how it's comparable to our AdoptAStream data now. But there is a level that we're calling the AdoptAStream action value. So that previous value, right, we're kind of thinking over the term of a year or maybe a longer period of time where we can kind of think about that 10% and what that looks like in terms of that 410. But if you get a result above the AdoptAStream action value, which is above 1,000 CFUs per 100 milliliters, then that's when you can immediately take action. We don't have to wait around. We don't have to think about doing any calculations, anything like that. If you go pull all of your plates out, you do the calculation to see what's my final level going to be or the result for this monitoring event, and it's 1,000 or anything above that, definitely want to take action. So the first step would be notifying your AdoptAStream local coordinator, who you should be able to be in contact with. We're going to help connect everyone if you don't already know who that person is, and contacting your local water authority and letting them know what you found. And then you want to go out, if it's possible for you to go out safely, and investigate that site and identify some possible sources of contamination. Because if you find a source, then it's going to be much more efficient for your local water authority and for other people to come out and mitigate that issue. So maybe you find a sewer spill or something like that. If you're finding exactly where that leak is coming from, or the overflow is coming from, that can be identified really quickly. Our local water authority can know, and they're going to come out and take action. So by you taking action, that's going to lead to others taking action, and we can make sure that we're stopping that source of pollution. So that's great. We hope you do find the source, even if it may be a little yucky. It's definitely worth it to go out and find it, because that's going to get these issues solved a lot faster. However, if you get this value, right, and it's 1,000 or above 1,000, you went ahead and notified your local coordinator and your water authority, you went out and looked, you didn't find anything, then what do you do? We recommend you take another sample to see, maybe was it a fluke, or maybe was it because something was just running down the stream that day. Because remember, like we said at the beginning, this sample is just a snapshot, right? So it could just be, kind of one little quick thing coming down. So sample again. So then we're going to compare what that second sample looks like in terms, or against what your first sample looked like. If it's below the threshold, then don't worry. You can just kind of follow up with your coordinator and let them know, I sampled again and I got, my normal level of 200 or 100 or whatever it may be. But if it's above the threshold again, so you're above that 1,000 level, definitely want to follow up and say, I can't find what's making the level so high, but I got these results again. We need to get someone out there to do some more rigorous monitoring and find the issue, which these local water authorities are really, really helpful in getting done. So, all right. And then we have safety. Jackie, don't let me go too far, but I think this is my slide as well. Safety. So safety is incredibly important in all AdoptAStream monitoring programs, but there are, especially some things in the bacterial monitoring that can cause potential harm. We've been talking about pathogens this whole time, right? And we've had people chiming in about wearing gloves and washing your hands. So making sure that that's definitely part of your protocol when you're going through is wearing that PPE, the proper protection equipment, when you're sampling, when you're plating, you're collecting, all that good stuff. That's a pretty important part of this. And then something we have on every monitoring PowerPoint is try not to sample alone. It's always good to have a friend, right? But it's always good to have a buddy when you're going out to a stream or maybe a secluded area. Just in case something happens, there's another person there to help you maybe get out of a sticky situation. Make sure you have safe legal access to your stream site. Don't sample during high flows or after a heavy rain event, and kind of that same disinfecting and all that good stuff. This is not a complete, full, extensive list of safety things. We have a more extensive list on our website, but yeah, just want to make sure that we're being really careful. And if you ever feel unsafe or uncomfortable, then just do not sample. We just want to make sure that everyone is safe and taking those proper precautions. All right, let's see. This is also neat. Okay, so once you're certified, you have made it through this presentation that Jackie and I are giving you, and then you make it through going through the exciting field portion and getting some practice plating with your local coordinator. You take and you pass the test because everyone's going to do amazing. So then what do? You will then get an account and get registered with our online database. So you'll have something there, you'll be able to log in and you can enter your data because only certified volunteers can submit data. We want to make sure that because your certification is only valid for 1 year, we're checking in with our volunteers annually to make sure if we have any changes to information like we have in this PowerPoint, that all of our volunteers are updated on that. And we're just making sure and checking in that the proper protocols are being completed throughout each monitoring event. So we'll just check in with you, make sure you're certified. Once you're certified, you have access to this database. You can enter data. There we go. So you're collecting the data, you're putting it in our database as soon as possible, I might add. So then what does the data do once it's there? Well, really, how your data used is completely up to you. We do, it is used for some things that maybe you aren't looking at, but truly what you're going out and doing for your stream and collecting your data, how you use it is completely up to you. These are just some examples of how data is used, but really it can be for student research, it can be for outreach opportunities, all different kinds of things. But one of the main things that we like to kind of talk about is what Jackie talked about in the beginning, is establishing some baseline conditions for our waterways. So establishing baselines is really important. It can tell us a lot, and it tells our regulatory people a lot of information too. If you're discovering maybe a sewer spill or something, when you're out doing your bacterial monitoring, you can report those issues and get those issues resolved, which is pretty gratifying experience, which is nice. And then education, you can help inform the status of streams for 303D and the 305B list. But also, we have that local or the annual conference every year. So if you have data that you've collected, you want to do research on it, you can present it there, all different kinds of things.
All right, and to wrap things up, we'll just walk you through the database login and a little bit of the data form entries. So this is the login homepage. Like Bailey said, once you complete your certification and you pass, then your trainer will contact you with the login information that you need to actually access your profile page. So we can go on to the next slide. All right, so this is the online data submission form. You have to be logged in with an up-to-date certification in order to see this page. And this is where you're going to enter all the different types of data that you collect. So you can see that there's a tab there for bacterial data. And we'll go over, this online form in a little bit more detail here in a minute, but we'll go on to the paper form. So all the paper forms have a very similar format to the online forms, and all of them also have this general site, weather, and observation section. So you'll want to make sure you put down the site information of like who's monitoring with you, the time and the date of your sampling, your group. Also take note of those weather conditions, because remember, observations is very key to monitoring. So weather conditions, water quality, water color, surface flow. Those are some key observations you're going to want to note for. And then moving on to the bacterial section, this kind of looks again like what the table looked like on our examples, but this is where you're going to put in the final results of each of your plates, your sample holding time, date, time, min, max temperatures, and we just ask that you submit your data from the paper form to the online entry form as soon as possible. So moving on, this is the online portion, and you can first put in the site, weather, and the observation entry notes, And then before you move on, though, to actually entering your bacterial information, you need to remember to click submit at the bottom of the page. That way it saves all of your entries, and it will allow you to move on to enter the bacterial portion. So yes, after you click submit, then you can go to the tab and put in your colony counts, but pretty straightforward. And then to wrap it all up, this is our slide. So you can follow AdoptAStream and stay connected with us. This is our small but mighty team of 4. We have our email there, website, Facebook, Instagram. Feel free to take a screenshot of all that information, but it's all available on our website as well if you need to get a hold of us